we are back. Okay, so DNA replication. That's right. So here we were. We just got to like second slide. That's something. Yeah. So the properties of the DNA structure help us figure out that it actually is going to help us with the replication as well, where each one of the strand can act as a template for the new synthesizing DNA during S phase. So initially, we didn't know exactly how this was going to work, right? So there were many models that were made for DNA replication um, for different, and predicting different outcomes. So the three main models that people worked on during that time were what we call semi-conservative, dispersive, and conservative. In semi-conservative model, the idea was that each one of the strands, so if you were to label them like it had in the other slide, S and S prime, each one of the strand will independently serve as a template. And so the newly synthesized DNA would contain half new, brand new, and half old DNA. In the conservative model, they claim that the entire helix acts as a template. So when you get your new template, it's either all new or all old. You don't have a mix. And in the dispersive model, they claim that you would have regions of the DNA that are going to be old and regions of the DNA that are going to be young or new. They are not all going to be made entirely of old or entirely of new DNA. Now, there were good reasons for each one of them. Each one of them were backed by some ideas about how and why they thought this to happen. Um, but obviously, that's not enough. You had to actually test it out. So, to test it out, the actual experiment that finally allowed us to figure out how it is that the DNA is synthesized was actually quite simple and cool, very quick and simple. So in this one, they took some bacteria, and bacteria is a perfect model system for this because um, they replicate every 20 minutes. Yes, so conservative DNA replication said that the entire double helix as a whole acts as a template. So the new DNA that is made is either entirely new, and then you have the entire old one intact. So at the end of the day, if you were to look at a conservative model, you could take apart which one is brand new and which one is old. You didn't have a mix of them. In the semi-conservative, it was that each replicated strand, each molecule at the end of the day would be half old and half new. Okay, so in this one, like I said, bacteria is a perfect model system because they replicate very, very quick. They took some bacteria and they grew them in media that contained nitrogen 15, and they grew another set of bacteria, another culture of bacteria in nitrogen 14. So this way, because they are going to incorporate the nitrogen in their molecules that they are building, they are going to be one or the other. They'll be labeled. N14 is going to be lighter. So it's obviously, if you run them on a gradient column, which for density, you will be able to see the difference. You will be able to see that the N14 band is going to run closer to the top of the tube, and the N15, which is heavier, will sink further down. So you could isolate those two populations. Okay, everyone good at that? Okay, so now they went ahead, and this is done by Meselson and Stahl. What they did was they now took bacteria and they grew that in light medium, and they got the results so they knew where it where it landed, and they look at it and they find that the DNA is all at a particular level. They did the repeat, and they found out where that band would last to. They pelleted the bacteria, so they spun them down, and they transferred just the bacteria, none of the heavy medium, just the bacteria, into light medium. And they continued to grow them. And now they looked at where the band, when they brought it down and centrifuged it, where they looked at where the DNA band went or where the bacterial band lasted. And they found that it came neither to the top nor the bottom location that the other two had gone to, but instead was somewhere in the middle. Well, that as such doesn't tell them anything more than that it's not conservative, right? Because in conservative, you should have had some that were high and some that were low. The here, you're getting just single band. So it tells you that it's definitely not the conservative model, and it's definitely that the new molecules are a mix of old and new. But it doesn't tell you how they are built. It doesn't tell you where it's one strand old, one strand new, or whether each strand is a mix of old and new, right? So these were then your two interpretations, two possibilities for further examination. So then there were other experiments where they looked at these a little bit more in detail by using further types of solutions in between to examine how they got the different replication. So in the end, what they found was that it is indeed a semi-conservative process. It's not that each molecule is a mix, but rather that each strand is either old or new, but the replicated molecule will contain one new and one old strand. So the original parent is going to have whatever, maybe it is completely not labeled at all. When you put it in a particular media, the new strands that are built are going to be half old, unlabeled ones and half labeled ones. Then when you add them to a different media, you will end up with now incorporating that new media into that as well. And that way you see that there is a whole variety of strands that are formed. So that helped them figure out that it is a semi-conservative process and not a conservative or a dispersive one. Any questions on that? So everyone gets that it is a semi-conservative process where each strand is either, each double helix in the replicated DNA is going to have one strand that's old and one that is new. Yes, no?
Yeah, that's actually pretty cool.
I know, isn't it? So yes. And usually what they did was they would put them, with nitrogen 14, then they would put them in nitrogen 15, and then they would put them back into an unlabeled, and they would see how quickly the label was dispersing away. And eventually you should have no label if it was a particular model. So it was pretty cool how they were able to, very simple experiment, and very reasonably quickly able to figure out which model was correct. Okay, so how does this happen exactly? How is it that this whole process is happening? So the synthesis of DNA, if you remember our DNA, for example, in humans, it's 23 pairs of chromosomes, right? 46 total chromosomes all over the place in there. Each chromosome will have multiple origins of replication. So synthesis begins at these set locations that are called origins of replication, which are where it's going to start to essentially melt away and separate the two strands. The double helix is going to get opened up. This happens with the help of initiator proteins that essentially separate the two strands of DNA and kind of open the zip up, so to speak. And then single strand binding proteins bind to that open strand to prevent them from coming back together and forming the double bond again, or the hydrogen bonds to form the double helix again. So the single strand DNA template is then ready for DNA synthesis, right? So you have these replication origin where the initiator proteins are going to bind, open it up, and then single strand binding proteins are going to bind and stabilize them in that single stranded open state so that they don't come back together and form their hydrogen bonds again. So here you can see that this is one strand, it's a portion of a chromosome, it's a portion of the DNA, not all of it, it's just a small area of the chromatin. And you see that it has multiple origins of replication. It's not that that strand has just single one, it's going to have many, many origins of replication. Each one of them, when opened up, will then start to replicate in both directions until they reach the second strand or wherever they meet. And that's how you are able to efficiently replicate the entire length of your chromosomes or the entire length of your DNA in a very reasonably short period of time. So our DNA can be replicated in an 8-hour period. That's how long our DNA takes. The bacterial circular chromosome DNA obviously takes very quick because their replication time is just 20 minutes. So obviously that's going super fast. But for ours, that's incredibly fast replication, right? At the end of the day, if you think about that, we have billions of letters in that DNA to copy, and it is able to copy it in a very short period of time. I think it was in your own book that it stated that it's like copying 1,000 of those books. That's whole entire, whatever the size of this book is, it's probably about 800 pages in a very short period of time, in 8 hours. Now, again, each one of these origins of replications will have their own complexes coming together, replicating the DNA in both directions until they reach, they meet, so that you can have that entire area replicated. So here you can see that more in detail. So you see the nucleosomes, right? So you see the DNA is still wrapped around histones. As it is going to start getting replicated, it will open up, obviously, so that the DNA can be copied appropriately. And you see multiple origins of replication forming throughout this area. Okay. The second thing to remember in this is that there is a directionality involved in DNA synthesis. So the new strand, because of the molecules that are going to come in and bind to the DNA polymerase that we'll talk about, that will come in and bind and actually read and copy the DNA, can only go in the one direction. So if you can compare it to reading an English language, you can only read in one direction. I can't read the opposite way. So same thing here, the replication can only happen in one direction. So it happens always in a 5' to 3' location. So that means for your template strand that is 3' to 5', there's no problem, right? It's just gonna, that's the 5' area. It's just gonna go ahead and it will keep copying. But when it is starting the opposite, the complementary strand would be starting at 5'. So for that one, there'll have to be a slightly different method in which to replicate and copy that direction. So now let's think about how it is that it is able to combine and make that complementary strand. So the molecule that does the reading and putting things together is DNA polymerase. The DNA polymerase looks like this horseshoe that's structurally. It has two separate sides. One side is the catalytic side to engage with the incoming nucleosides and connect them to the strands to actually prepare that complementary strand of DNA. The second side of this horseshoe is meant for proofreading, and we'll talk about that more in detail in a few minutes. So if we were to look at our DNA again, this little strand of DNA, in the 3' to the 5' location, that's your template strand. It's always going to be the 3' to 5', because the new strand can only form in the 5' to 3' direction. The new strand is going to form by the DNA polymerase by getting the appropriate nucleoside triphosphate. Now, this nucleoside is triphosphate. Remember, it has 3 phosphates attached to it, just like adenine triphosphate. So we've seen those. So it's a nucleoside with triphosphate. It could be adenosine, it could be thymine, it could be any one of them. And then when it comes in, the appropriate one that can bind to the opposite side, right, that can interact appropriately with the hydrogen bonding. So A with T, C with G. will be the only one that would stabilize that interaction. At that point, it is going to release a pyrophosphate. So it's a catalytic reaction. It requires energy input, and not just one, but two phosphates to be broken off. And while this bond is getting made, right? So the 5' end of the incoming nucleoside will attach to the 3' hydroxyl from the one before. And that is how you will create that, grow that strand in the 3' direction. So again, it is an DNA polymerase is an enzyme, but it is requiring input of energy in the form of these triphosphate nucleosides that are coming in. It's removing two of those phosphates in the process as it binds stitches these two together, the two nucleosides together. And it's not the nucleosides that are forming the bond, they're forming the hydrogen bond, but the actual covalent bond that's happening is in the sugar phosphate backbone. Okay. So like I said, that's very easy to imagine for your template strand that is going already from the 3' to the 5' location. However, let's look at the opposite side, the complementary strand on the top. That one, when it starts to open up, if it was the beginning, if it wasn't an open-ended loop. So remember, the origin of replication are kind of just, opening it in the middle. They're not opening the corner. But so there will be an area that is going to be in the opposite direction, the 3' to 5' location. Again, the origin of replication has to be opened enough for the DNA polymerase to bind to the 5' end of that template strand and then grow it in the 3' direction, again, the same way. Okay, so as it is opening up, so if you look at this DNA fork, let me get, so as this gets opened up, on this side, it's just going to continue to make this strand. The more it opens, the more it's able to make it. On this side, it has to, it is making the strand in this direction, in the opposite direction. So it's going to continue this on this side, but when it continues to open up on here, it has to wait until it has opened enough so that it can go back and then start building again. 5' to 3', because that's the only direction that it can move. Okay? Yes, no, maybe?
Yes. And each strand will have its own DNA polymerase?
Yes. Okay. Yes, they have their own complex, the whole thing. It's not the same. It's many DNA polymerase, or actually the whole initiation complex, everything. There'll be multiple ones of these all along your DNA at each one of the replication fork. So the one that's working on this side is not the same one that's replicating over there. There's a second one over here, right? And there may be another one that comes in, or this may fall off and then go back and replicate here. But it's going to be multiple complexes that are walking around the DNA. That's why it is able to do it in such a quick manner.
Yes, got it. Thank you.
Yes. So a few just reminders. The DNA polymerase cannot synthesize DNA all by itself. So it can't just latch on the DNA and start by itself. It requires some kind of a nucleic acid portion to hang on to bind to, in order to replicate from that point of origin. So that little piece or hanging piece that it is required to bind to is provided by primase, which is another enzyme. And its job is to produce an RNA primer that is complementary to the template strand, so that it's just long enough for the DNA polymerase to bind to it and initiate DNA synthesis. Otherwise, DNA synthesis cannot start because the polymerase can hang on to the template strand alone. It requires a double strand latch-on point, so to speak. So the DNA polymerase will then bind to the primer and then build from there. Now that causes a problem because now you have these little primers along the DNA strand wherever the new synthesis has occurred. Well, those RNA primers are then later removed and replaced with DNA to catch it all together and to stitch it all together as one long DNA molecule, the way it should be. So here is an actual, before we just saw one side of the replication fork. So here is an actual origin of replication, the way it would normally look. Again, both sides of the strand. So if you think about this as your origin of replication, if this is where it started, right? In this case, you can see this would have been where it started to open up. And so the 5' to 3' direction just continues straight, straight, straight. However, at this point, the DNA polymerase could not have bound to the 5' end because it was the wrong orientation, right? This 5' end, because the DNA polymerase has to work 5' to 3'. So it had to wait for the origin of replication to open enough so that it could start building its first strand. And it would have done so, right, until it reached another strand and fell off. Then in the meantime, another little portion would have occurred. So then the polymerase would bind further back and continue, and further back and continue. And so there'll be a bunch of small fragments of newly synthesized DNA along this side of the arm, the 3' to 5', on the 3' portion of the origin of replication. And these little fragments are what we called Okazaki fragments, after the person who found them. Now, it's not like it's only present on one side. The same thing can be seen on this side as well, right? So here as well, because this was the original origin of replication that continued, the ones that are starting from here are, again, in the same conundrum, and they still have to then open up every time a little bit before it can continue the new strand, okay? So the leading strand is the strand where it is the replication is happening in a straight, non-stop way from 5' to 3' location. And the lagging strand is the one that is called lagging because it has to kind of slowly catch up as the replication fork gets extended in small fragments, Okazaki fragments. So for your 3' to 5' location, your leading strand is going to be in the front and your lagging strand is going to be on the back of the replication fork. And on the complementary side, on the 3' strand of your DNA, it's going to start off with the lagging strand and then get to the leading strand on the left-hand side of the fork. Okay? Yes?
So is the lagging strand always on the 5' end of the newly synthesized DNA strand?
So the lagging strand is always going to be on the 3' end of the template strand, if that makes sense, right?
Is it because like the DNA polymerase needs to recognize this part of
It needs
opening?
enough openings to bind so that it can create the new strand from 5' to 3' orientation, because it's always going to grow.
So that's why it's lagging.
It's lagging because it can't bind to anything until it's opened enough. Yes. So the area where you have these small little fragments forming, the Okazaki fragment forming, which is always going to be on the 3' part of your template strand, that part is going to be the lagging strand. And so in here it's showing you in a typical example where you have a 5' to 3' on top and 3' to 5' on the bottom, In that case, the lagging strand template is going to be on the right side of the fork for the top strand, and on the left side of the fork on the bottom strand. Okay. Basically, just look for the 3' end. Whichever side of the replication fork is the 3' end, that's where all the lagging strands are going to be. That's where Okazaki fragments are going to be. Okay. Okay. It's raining cats and dogs outside. Okay, so let's look at how exactly the primase adds that primer, the RNA primer, in the beginning so that you can have a DNA polymerase bind to it. So here is your template strand. It's already opened up. It's already probably stabilized with the single strand. Well, it would be definitely stabilized with the single strand binding proteins. The primase is going to come in. And if you look at the shape of primase, it's very different, right? It's almost like a hook rather than a horseshoe like you saw with the DNA polymerase. This primase is able to only bind to ribonucleotides and not to deoxy ribonucleotides. Again, these are going to be ribonucleosides, triphosphates that will come in. It will do the same exact way of stitching together a short primer based on the sequence that is right there to create about a 15 to 20 nucleotide primer, and then it would fall off at that point. That gives enough of a hangover essentially for the DNA polymerase to come in and bind. The primase also synthesizes in a 5' to 3' direction, but it doesn't need, it can hang on to a single strand of DNA. It doesn't need 2 DNA strands to be able to bind to it. So this allows the DNA polymerase to have a latch to hang on to get stabilized with. Now, when we get to both the leading and the lagging strand, there are going to be different enzymes that are going to be required to create that whole sequence. The leading strand is easy because it's just going straight without any broken, any nicks or any smaller fragments. So for that one, DNA polymerase, once the initiator complex is formed, once the primase has added that initial primer, and once everything else has been brought together to create that replication complex, the leading strand will continue to read on and replicate until it reaches whatever end it needs to go to. However, on the opposite side, where you have your lagging strand, you're going to require some additional enzymes now, because now you have to figure out a way to stitch these small fragments together. And now you have a lot of little primers all along the way made of RNA instead of a single one in the beginning of the leading strand. So for these ones, you're going to have additional needs for that to become appropriately stitched long strand of DNA, complementary strand of DNA. So in this case, once the DNA has replicated, what happens is that you have the primers are removed, the RNA primers are removed, and the DNA that was already done before in that previous, right? So you have these little Okazaki fragments, but now the DNA polymerase does have a little place to hang on to. It can bind to this and it can fill in the gap until it reaches to the next Okazaki fragments DNA portion. So that's essentially what it will do. The Okazaki fragments are removed, so you have these little gaps, and then that it's filled in by a repair polymerase that works very similar to the DNA polymerase, but will bind to the DNA itself and then use it as a latch-on place and then go from there until it reaches the next portion. You'll still be left with these little nicks, these little breakpoints that are then needed to be sealed. And these are sealed with the help of another enzyme called DNA ligase, which is essentially going to stitch those bonds together. Its sole job is to fix that one bond right there at that stopping point. when the 3' hydroxyl and the 5' sugar. That's the total thing that it's going to do. Okay. So DNA ligase, and this is again, now it's going to show you how it works. DNA ligase joins the Okazaki fragments. This time, remember, there's no complementary base pairing because all the bases are already there. It's only the sugar phosphate backbone that needs to be fixed. However, it still needs energy input to make that bond. Before we took that energy from the incoming nucleoside, which came in as a triphosphate, so you could use 2 of those phosphates. In this case, you don't have any nucleoside coming in to be part of the DNA itself, but you have ATP, right? You have adenosine triphosphate. So you're going to use ATP as your energy source, like you typically do, hydrolyze it to, again, similarly, where a pyrophosphate is removed, and that ATP is then used to join together those bonds to make a continuous strand of DNA. And the adenosine at the end of the day, as a monophosphate, just with a single phosphate, will be released back so that it can then again be converted back to ATP. Okay? So do you get this process?
Yes.
Right? So whether the DNA is using the complementary base to create the DNA, to stitch together the new DNA strand, or whether it's just to stitch a nick, it always needs that some type of a nucleoside triphosphate to come in, that then they can release the pyrophosphate, use that energy to create that bond, and then either they're going to incorporate that nucleoside at that point into the base pairing, or they're going to release it in the form of AMP so that it can be converted back to ATP. Okay. Now, remember that horseshoe shape we talked about in the beginning for the DNA polymerase, and we talked about how that was because it had 2 different functions. So the front portion of the DNA polymerase is doing the actual putting together the complementary strand. The back portion of the DNA polymerase, the second part of that horseshoe, is proofreading its own work. So there is a built-in kind of accountability within the DNA replication process, which is why we are able to stay the way we are and to maintain our integrity as an organism. It is a very important function that it also holds in addition to it's just putting together the DNA. So let's say that, here you have the DNA polymerase, it is working together and it accidentally has gone ahead and stitched the wrong nucleotide to the DNA. Well, that incorrect pairing is not going to work because the hydrogen bonds are not going to be made properly. Or it may be that it's put 2 pyrimidines together and now the size is not correct, right? Because that would be 2 double ring nucleotides together. In that case, because the base pairing doesn't quite work, it causes essentially a kink either in one direction or another that shifts this the positioning of this enzyme to the back location. It recognizes that, that's how it's essentially recognizing that it's not able to move forward correctly. And so that nucleoside would then be removed, and then the new one will have to be put in. Correct one will have to be added just like before, so it can continue on with the rest of its job. Okay? Questions? Yes, no.
I had a question about the beginning of that kink that happens.
Yeah.
So the rest of the DNA is still attached with a kink in it, or is it just
so
now?
It's already connected at the backbone, but it's unable to shift into its final position because the hydrogen bonding is not there because of the, incorrect pairing. Does that make sense?
Yes, thank you.
Yeah, so it's essentially, like with those bead necklaces where we kind of push them in, it's just not going in properly. It's just not gonna stabilize that bond. So because that bonding doesn't happen, it is unable to shift in the final position where the polymerase can move. And instead, it will shift towards the backside of the polymerase, which has a catalytic unit, its own little active site. So the same polymerase has essentially two possible active sites, right? One, which is getting the nucleotides and nucleosides and putting them together, stitching them together into the DNA, and one that is recognizing the mistake and removing that problematic nucleotide. Okay, so here you can see those separate sites in this particular model. You have a polymerizing side and you have the editing side of your DNA. So when it is polymerizing, it is just interacting with this, and this back end is not incorporating in anything at all. It's just kind of hanging on there while this is moving along. However, as soon as that interaction destabilizes because it's unable to form the appropriate bond, that shifts the DNA strand essentially into the editing, because it can't keep moving forward. So it shifts towards the back to that editing active site where it is then removed and then continued with its appropriate strand. Okay. Okay, so next, let's think about all those other proteins we talked about. It's not a one-person job to replicate DNA. There are many, many other proteins that are involved in that initiator complex, and then to maintain that open single-stranded DNA structure so that replication can occur. We already talked about single-strand binding proteins. They essentially bind to the strands, the single-strand DNA, when it's opened up, so that they don't come back into their typical double helix, or the base pairing cannot happen. Now, before that, obviously, you had to unwind the DNA. And the unwinding of the DNA is done by a particular enzyme. Its sole job is to unwind the DNA and make it into that straight DNA molecule that can be separated, and that's DNA helicase. The DNA helicase is disrupting the hydrogen bonds between the bases so that they can separate at the origin of replication specifically. So the origin of replications have specific sequences that allow the DNA helicase to bind to them and start to pry them open right at that spot. It can't just bind anywhere along the chromosomes or the DNA. It has to bind in specific areas where it can bind to those origin of replication sequences. Now, the other thing that we have to think about is how to prevent the DNA from breaking apart as it is unwound and as it is replicated. So if if you think about it, it is a double helix. It's twisted, right? It's tightly twisted in this helical form. When you unwind one portion, you're obviously going to cause more contortion on another adjacent section. And that can lead to bonds getting broken in the double helix backbone especially. So the topoisomerase is another enzyme that works to kind of distort, open up those distortions and relax the DNA so that it does not break unnecessarily. Now, topoisomerase itself actually causes nicks in that backbone to release the tension, but it does it in a very controlled manner rather than allowing the DNA to break on its own at all over the place. Okay, so this table summarizes all the enzymes that you guys should know about, all the components of this DNA replication machinery. We're going to talk a little bit more about some of these in the upcoming slides and what function they perform in each case. So here, for DNA topoisomerase, you can see that that's how it's relieving the tension, is by causing a transient nick in the system so that it can unwind that contortion that has happened ahead of the helicase. and keeps it from getting messed up. And then there are other additional enzymes that are needed in that initiator complex before helicase can unwind the DNA. This includes a sliding clamp and a clamp loader. So the clamp loader works with the sliding clamp to come in and gets, it loads onto the DNA double strand and binds to the beginning of the origin of replication after DNA polymerase can binds to the template strand. It stops the DNA polymerase from falling off the DNA strand, especially at the ends of the DNA, where it would just not be able to continue synthesis unless it has some place to hold on to and kind of stay in place. Okay. So here you can see some of these in function. So you have your DNA helicase that is opening up the DNA, unwinding the DNA. As it unwinds the DNA, you will have the single strand binding proteins binding to that lagging strand to keep it open and not prevent it from coming in back as a DNA double helix. You will then have primase putting in an RNA primer that can then be used to replicate the DNA with the help of DNA polymerase. And right here is the clamp, the sliding clamp that I talked about. It's holding the DNA polymerase and it's shifting it forward as it goes to continue replicating the DNA. Here again, you see the primase putting in a primer, right? That's going to then be replicated by the DNA polymerase, another DNA polymerase that will come in and bind to it and replicate that portion. So already you can see there are multiple DNA polymerase, machinery, this whole replication machinery joined on a single replication fork. It's not a single man's job. On the leading strand, you can see it's a little bit simpler. You just, once the DNA polymerase binds, once that initial primer has been made and the DNA polymerase binds to that leading strand, with the help of the sliding clamp, it just can continue until it reaches the end of that fragment all the way through. But on the lagging strand, the single strand binding proteins become important because the DNA has to remain open until the polymerase binds to that primer made by the primase and start the replication process. Okay. So here you see up close of this procedure, and you see both the leading and the lagging strand, and you see how this process has to, they all have to kind of work together for this process to work efficiently. So there are times where the replication, the newly replicating DNA on the lagging strand would bend over so that once it reaches, this polymerase reaches the end of one strand, it can hop over to the start of the next Okazaki fragment as well. There is an animation that is built in here. You can, if you click on it, it should take you to to a YouTube video that I like. So you can look at that as well. What time is it? We were gonna watch it, but since we are probably running low on time. Let's see. Where are we? Oh, we'll watch it. What the heck? We shall watch it. Well, watch my computer have a meltdown again. Oh, come on. Yes. So I will have to see what my computer's issues are after our class. Oh, it's working this far. Good. Okay, so let's now think about those Okazaki fragments, right? We have all those little Okazaki fragments that need to be stitched together. Well, that's great. We can go ahead and do that for all the internal portions of each chromosome. If you have a circular chromosome, there's no problem. You can get everyone in, no issues, because you always have a DNA or an RNA portion to hang on to. If it's a linear chromosome, now we have a problem, because at the very end of this journey, you are going to have the end of the chromosome with a RNA primer on it. And no way for DNA polymerase to bind and fix it. All these other ones internally are going to get fixed up. They're going to get taken over by that repair polymerase and they will take care of it. But this little end, right, doesn't have a DNA fragment to bind to that it can use as a place to initiate its replication. Now remember, DNA polymerase, the one for replication, cannot use DNA as a primer, as the beginning, right? The repair polymerase, on the other hand, can use a DNA as the primer, which is why it is used to fix these RNA primers, the gaps that are formed from them. Now, in this case, the RNA primers are still going to be removed, right? And they're going to be replaced by DNA all over by ligase controlling the next, fixing the legs. But there'll be a little bit of lagging strand DNA that's going to be hanging over with no replication in sight. Now, our bodies have a way to fix that in the form of another enzyme called telomerase. So telomerase can replicate those ends of the eukaryotic chromosome, but they are very specific on what they can fill it up with. They can only fill it up with a particular sequence, a particular set of sequence associated with their biology. They have their own little RNA template that has a sequence, and that repetitive sequence is the only thing they can use to fill this gap up. Good thing for us, our eukaryotic chromosomes have that same sequence in a very, 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 very long chain. They just have that repeated many, many, many times over, thousands of times over. So that's great. And in that case, that telomerase uses its bound RNA template that it is bound with to add the same repetitive sequence to extend that template. And then again, the same way, the DNA polymerase can fill it up. The ligase can fill the next. Life is good. Well, there's another catch. Telomerase, it's not active in typical somatic cells in our body. So it won't do that. So what happens instead is that these single-stranded ends get chewed up. And over time, the telomeres that are these ends of our chromosomes that have this repetitive sequence get shorter and shorter and shorter, leading eventually to what we call the aging response. Eventually, our telomeres get so short that now you cannot replicate without losing important DNA information. And at that point, the cell switches into a senescent mode and eventually dies out. That happens at a cellular level and at an organismal level, because as our ability to replicate is reduced with age, so is our body's ability to fix itself, and eventually then that's how you get your aging response. Okay, so questions about that? So this last thing actually just talks about how if you don't have a way to complete that telomeric double-stranded DNA, there's going to be a specific process through which the ends get looped over and this 3' overhang is cut out of that system, out of our cells. So this actually shows you how telomere lengths are vary not only by cell type, but also with age. So if you look at most mortal cell lines, you will see that they have a particular size of telomere and that it reduces with age as you become older and older. So mortal cell lines, the ones that are, going through it, they're able to maintain, they're able to, they are losing their telomeres with each cycle. And that's what's seen right here. It reaches a particular stage where now what it is in what we call a crisis mode, and it can no longer replicate the DNA. And still stay alive in its particular stage. Immortal cell lines actually reactivate their telomeres sometimes. That's one of the ways sometimes that they are transformed, is that they have their telomerase switched on again so that they can add those ends back into their chromosomes. And then even though they're old and damaged and not appropriate for growth, they can continue to grow. Okay. And so this is how you see that telomeric DNA at the ends of your chromosome reducing.
So, like someone can know how long he's gonna live if he can do like any test to show how much telomerase he has.
Yeah, so so you see that majority of the chromosomes in this beginning at the first part, at the very first cell, if you get them at an early age at the embryonic level, they're gonna have this really high telomere length, and then it stays stable throughout. By the way, throughout utero and utero development, telomerase is switched on. That's why we are able to develop as an actual organism. Otherwise, we die out before we even make it out. But once, as soon as as we are born, that telomerase is shut down in majority of our cells. So then you start to see that decline of telomere length. And there's a variability. You see the variability. That's why they're fuzzy. And then it kind of stabilizes unless it becomes activated again. And then you'll see a little bit of that increase to maintain that length at a particular level, like in this case, when the immortal cell lines. But yeah, that's one way you can look at how, kind of old your cells are. And how, how they have been taken care of by us as well. Questions? Oh, we made it in time. How did that happen?
So is the end of the chromosome the only thing that can stop replication, or is there like another like enzyme or protein complex that can stop it?
So you can definitely stop replication at any time. There would have to be a certain signaling pathway activated, maybe because there's no more nutrition or whatever, but usually what happens is the replication still goes on. What happens is the next step doesn't happen. The cell doesn't undergo mitosis, it hangs on in that G2 multiploid state until the conditions required to actually divide and make more cells. Our hepatocytes, for example, they are typically in a multiploid state, a lot of them. So they've already gone through synthesis of DNA and they are ready to go to divide as needed, but they're not dividing. It's because DNA, liver cells get damaged a lot, right? Because that's what they do. They detoxify cells and detoxify our body. And in doing so, they are getting damaged and getting a hit. So if there was a mass injury on these liver cells, that would trigger the response for these cells to go ahead and divide and make more cells to replenish the supply. Otherwise, even though they finish the synthesis of DNA, they won't keep going and they won't finish the cell cycle. So we'll talk more about what controls whether replication gets done all the way through or whether cell cycle gets done all the way through a little further, I think in a couple of weeks when we start talking about cell cycle.
All right, thank you.
But typically, if synthesis has started, it will usually go all the way. There are inhibitors that can stop the synthesis and kind of catch all the cells in a particular stage. But typically, if it has started, it will finish, but then they won't move to the next stage. Okay. Okay, guys. So that's all for today. Now, next class, we are talking about, we are doing transcription, right? But also we are talking about a chapter in our genome book. Where is my book? Right here. I think we are doing chapter 1, chromosome 1, life. So please do make sure to at least read the chapter before you come to class next time on Thursday, because we will have a little discussion group on it. So we will be, I have some points I would like to point out. I'm hoping, I didn't do that the first with the first chapter because it was kind of simple and easy, and we were just getting used to each other in the class. But we're going to start incorporating these discussion groups, and hopefully that's going to help you with those critical analysis questions as well. So we'll have a little bit of talk about the concepts that they talk about in that chapter called life. They give an alternate theory of what came first, humans, eukaryotes or prokaryotes, and they talk about what came first, DNA, protein, or RNA. And they have good arguments for what they suggest.
I already read that and I love it.
Isn't it fun? So we
Yes.
will discuss those alternate theories, and some of them have good scientific basis in them. And some of them are still just theories and still need further proof. So that is something we will talk about. And I would like you guys to, go through it beforehand so we can have an active discussion. Okay, guys, that's all for today. I'll see you.
I have a question.
Yes, Alicia.
Oh, it's not dealing with lecture part. It's dealing more so with the lab. I didn't get participation points for attending the Zoom on Wednesday, but I attended the Zoom last week Wednesday.
So just send me an email about it so I can double check. Sometimes because I get your attendance for lab from the Zoom attendance, your names show up differently sometimes in the Zoom, and I have to kind of use imagination to figure out who it is. Sometimes it's quite easily figured out, sometimes it isn't. But yes, I'll double check, no problem.
Yeah, that happened to me too.
So yeah, I think it should be okay. But yeah, if you have any problem like that, just send me a quick text. Also, my computer, same computer that's dying, the one on it is sticky. So sometimes I just type like 10, 10, 10, 10, 10 quickly. The one doesn't take, so it shows up 0. And then, but I have record of all your attendance separately in an Excel sheet too. So I can easily just double check and say, oh, that was my typo because my one didn't press.
That's fine. Thank you.
You're welcome. I'm getting a new computer, so I'm happy. Take care.
Have a good day, Professor.
You too.